Hello, Dr. Simpson and Dr. Raubenheimer. So you are both professors at the University of Sydney with a focus on nutrition and health. So you developed the idea of nutritional geometry and have published two books related to this, most recently, Eat Like the Animals. So welcome to Modern Healthspan, and thank you so much for joining us today.
It's a
Thanks,
pleasure.
Richard.
So I first heard of your work when I was talking with Dr. Lindsay Wu about protein requirements. And it sounded very interesting, so I had to follow up. And it's great to have you on the channel to talk about it. So, and I really like the rigorous way you look at the nutritional requirements and kind of build up from there. So you developed a way of thinking about these requirements called nutritional geometry. So could you explain what you mean by nutritional geometry and some of the background to that?
Well, over to Dave first.
Okay, nutritional geometry. When we got into nutrition, there was a strong, there still is in some respects, focus on individual nutrients as the drivers of nutritional outcomes. And we both came from a biological/ecological background, and we realized that actually the logic of biology is that nutrition will operate as mixtures. Nutrition is mixtures. So we thought, well, how can we develop an approach to understanding nutrition that's based on mixtures? And nutritional geometry is such an approach. So rather than thinking about how does protein versus fat versus carbohydrate influence, say, an animal's health or a person's health, we think about how did the mixture of those things influence the health, and what is the relative contribution of those different components? So it's more of a systems approach to nutrition.
And we first met in 1987. David arrived at Oxford from South Africa, ready to do a PhD, and he and I ended up together leading a first-year biology course in a medieval woodland outside of Oxford called Whiteham Woods. And we were supposed to be helping the first year students sample insects and the invertebrate fauna of the woods, and instead we got talking about nutrition. We got talking about how we felt that the science of nutrition and biology was a bit of a poor cousin. A lot of biology concentrates on sex and death, and not as much as it ought to on nutrition, which is the other really significant part of the triumvirate of factors in biology. And by the end of that, we'd come up with a plan, and that plan started with locusts and studies on insects, and has led us over the last 37, 38 years to where we are now in this extraordinary place called the Charles Perkins Centre at the University of Sydney. So it began with biology. It began with fundamental interest in why animals eat what they do and how they respond to the things that they eat. And it's taken us now to, we think, a really fundamental new understanding and a way of thinking about nutrition and health.
Right. And so I saw in your book that one of the things is you gave different mixtures to animals, and they seem to be able to determine what mixture of the three macros, the fats, the carbs, and the protein, that was optimal for them, or within the situation that they had, I guess, is also there too. So could you talk about what you did within the nutritional geometry in terms of how do animals decide that, this is the mix that I need?
So the fundamental understanding that we arrived at quite quickly was that we tend to think of animals as being hungry or being full. So they have appetite, and appetite has generally been framed around energy and energy requirements. And we discovered pretty early on, and we were among the first to do this, that animals don't have a single appetite. They have specific appetites for key nutrients. And in our studies on locusts, we showed that they have a specific appetite for protein and a separate appetite for carbohydrate another for fat, and yet others for salt and micronutrients. And we realized that these appetites have evolved to help the animal make appropriate food choices amongst available foods and serve to guide the animal towards an optimal point in a multi-dimensional nutritional space. So these appetites help solve the mixture problem. If you pick foods that have appropriate mixtures of nutrients, you can balance your intake of more than one thing at once. And these appetite systems help to do that. But
which is not surprising. Sorry, Steve, you go.
No, keep going, Dave. Yeah.
I was just going to say, which is not surprising because it's not food that the body needs, and it's not energy, exclusively energy that the body needs. The body needs different nutrients for different purposes and at different amounts in particular times. And as biologists, we thought, well, how has evolution solved this problem? And we predicted that the body would need to be able to tell the animal's brain what it needs at what particular time and how much of it. And that is what nutrient-specific appetites do. So our experiments with locusts basically demonstrated what we expected from basic biological and evolutionary theory to be the case.
And that allowed us to then delve into the physiology, the mechanisms of these specific appetites. And some of those are really beautifully elegant in insects. So, for example, the way an insect tastes its food depends on its requirements. So if it needs protein, its taste receptors respond really vigorously to amino acids, which are the signal for protein in food. And when the animal doesn't need protein, its taste receptors don't respond at all. So they're blind to protein, and hence the animal won't eat foods that contain what it doesn't need and will specifically choose things that it does. And we showed that they can learn to associate particular cues with the nutritional composition of those foods. And these mechanisms are really powerful ways of the animal navigating this complexity of nutrition. So that was all well and good. But then what we did next was to put locusts and other insects in worlds where they couldn't mix. They were stuck on a particular composition of diet. And under those circumstances, you force these specific appetites that would otherwise work together to compete. And when we did that, we discovered that the protein appetite in our locusts was particularly strong. It would dominate the other appetites and lead to the locusts eating either too much or too little of everything else to ensure that they got the right amount of protein. And that was a fundamentally important insight.
Right. So if I'm not getting enough protein, then I will eat more carbs just to, even though it's going to make me fat, because I need that much protein. So
it's a little bit like getting the same amount of protein from, say, a protein-rich food like meat or fish versus from a donut. Now, those insects were stuck on one of those two, equivalent of one of those two alternatives. And on the donut, they would have to eat much, much more food and much, much more fat and carbohydrate to get the same amount of energy as they would at a much lower intake on a protein-rich food.
So a couple of questions on that. So do the animals sense all amino acids? I mean, like mTOR specifically only senses like leucine and arginine. So is that the same? Is it specific amino acids, or do they sense like all of them?
No, that's actually a really important question. So how does the protein appetite system respond to amino acid balance remains unresolved. So you're right, some of the signaling systems that we know about, there are nutrient sensing systems for amino acids that have overlapping sensitivities. Like you say, the mTOR pathway has a particular predilection for the branch chain amino acids, but there are other amino acid signaling systems. How that actually operates is not yet fully understood. It's a really active area of research at the moment. We know that circulating amino acid levels are important, and they're detected at various stages, not only in the mouth and the gut, but internally as well, in various places around the body and ultimately in the brain. And we also know that the protein status is translated into hormonal signals, one of which is now increasingly well understood to be a protein hunger signal, and that's fibroblast growth factor 21, or FGF21, which is largely produced from the liver, acts in the brain, and has a sort of coordinated set of effects on behavior. It increases the liking for savory flavors, so it's a protein appetite signal. It'll decrease the liking for sweet, or the seeking of sweet flavors, if the animal is commensurately high in carbohydrate. And it also activates various other metabolic responses. So it's a bit of an orchestrator of protein appetite.
Interesting. Have you seen, so for locus, protein is the kind of the deciding factor. So across the different animal species, did you see different deciding factors for how much should be eaten?
We saw quite a wide variety of responses, but far and away the most common one was this protein prioritization pattern that we've discussed. So work has shown, for example, on wild primates, our closest living cousins, that most of them do the same as what we do, is regulate most strongly around protein, but there are a few interesting little exceptions that don't.
If there's one mineral you should be worried about not getting enough of, it is magnesium. Magnesium is the body's master mineral, powering over 600 critical reactions, including hormone balance, stress management, sleep quality. Even digestion is influenced by the presence of magnesium. But there are two big problems here. The magnesium in the soil has been drastically reduced since the 1950s and is also removed by food processing, which explains why it's estimated that up to 75% of the population may be deficient. And most supplements contain only 1 or 2 forms of magnesium when our body needs 7. The only magnesium supplement on the market that has the full spectrum of all 7 forms is Magnesium Breakthrough by BiOptimizers. BiOptimizers are offering an exclusive deal for my audience, and it's for a limited time while stocks last. Right now, you can get a bottle of Magnesium Breakthrough for free. Just go to magbreakthrough.com/modern free. Thank you for your support. So you did some, kind of moving on, you did some mouse experiments. I think they were mostly mouse experiments, where you were mixing and matching the 3 different macros, and you saw like trade-offs between health and, well, maybe Healthspan and lifespan and the ability to have offspring. So could you talk about those experiments, and what did you see as kind of the trade-offs that the animal had to make?
Well, the really fundamental, it's a couple of things are important here. One is why we decided to do that, and the other was how nutritional geometry provides a way to do it. So let's consider both of those. Having established that there was a protein-specific appetite and that it was universal from slime molds to wild primates, as David said, it begs the question, why do animals not only care about eating too little protein, but also apparently about eating too much? So they regulate to a sweet spot. They don't eat too much and they don't eat too little. And this protein leverage response, as we call it, indicates that there must be some cost whatever that happens to be, adaptive cost, anyway, evolutionary cost, to eating too much as well as to eating too little. Now, we understand what the cost of eating too little protein is. You can't grow, you can't maintain your tissues, you can't reproduce. But why should there be a cost to eating too much? And to answer that, the way to use nutritional geometry to answer it was to confine animals to one of a large number of different mixtures of protein, fat, and carbohydrate, and to measure what happened, and to map what happened, and to visualize and analyze those maps to try and, quite literally, it's the cartography of response to nutrition. And we initially did that in flies because fruit flies are cheap and short-lived, and you can, as we did, take 1,000 female flies and put them on one of 28 different diet mixtures and then measure how many eggs they laid and how long they lived within a reasonable period of time. And the experiment takes up a tabletop rather than an entire factory, which is what you need when you pick a bigger and longer-lived animal. And the answer came back really very clearly that in these flies, they lived longest on a low-protein, high-carbohydrate diet, but they produced most eggs on a higher protein, lower carbohydrate diet. And on a very high low-carb diet, they neither lived very long nor laid many eggs. So the longevity and the reproductive landscapes in nutrient space were different. So high protein was better for reproduction, but it traded off against how long the animal lived. So that was well received. It was a paper that had a big impact when we published it, but begged the obvious question. People would say, well, that's all well and good, but they're flies. What happens in mammals? So then we set about to do the equivalent experiment in mice. And that involved, rather than a tabletop, 5 years of research, 6 tons of diets, which we varied in protein, fat, and carbohydrate, the 3 macro dimensions. And essentially, we found the same result, that the animals on the lower protein, higher carbohydrate diets lived longest and were most metabolically healthy in later mid to early late life, whereas those on a higher protein diet had greater reproductive function. So it was a similar pattern to what we'd seen in flies. We've since gone on and we're currently analyzing this at the moment to do an even bigger experiment where we're measuring the impacts across the life course of being confined on, and again, one of 20 diets in this case, in 2,500 mice. And we're finding exactly the same results as we found in the earlier study, but now we're able to track how requirements are changing across the life course. And that, of course, is a really important part of the story. And we can elaborate that in a minute.
Yes, I definitely want to talk about changes in protein requirements. So in the low protein, high carb diet, the animals lived longest, but they were a bit chubby, is my understanding. So the lean, healthy ones were on a higher protein diet. So do we know, why did the lean, I guess the lean ones, I don't know if they're healthy, the lean ones, why did they die? Was it any particular reason?
Yes. So what, you're quite right. They made good looking corpses and they died earlier. And when we looked at the age-specific mortality across the life course, these animals were put onto one of these diets at very young age when they were just weaned and left throughout life unless we sampled them at a given age in mid-late life to measure their metabolic health. They were left to live to the end of their life. And it turns out that the age-specific mortality during the particularly early life up until late life was higher on the high protein, low carbohydrate diets than it was on the low protein, high carb diets. But if mice survived until later life, till, old age as a mouse, then they survived better on a higher protein, lower carb diet. So what it was telling us was that the higher protein, low carb diets were having a higher risk of mortality, particularly in the middle age grouping or part of the life course. And that was associated with a whole range of different markers, all the traditional markers of the biology of aging, telomere lengths, mTOR activation, IGF-1 levels in circulation, etc., were higher on the high protein, low carb diets during that sort of middle age period than on the low protein, high carb diets. Things like tumors and rates of various cancers and issues that clearly are life-shortening, they were also elevated. So broadly speaking, metabolic health, insulin resistance was worse and so forth, was worse on the high protein, low carb diets. But you're exactly right. The healthiest, longest lived mice were a little bit fat, and that's because mice also show a degree of protein leverage. So on the low protein diets, they were eating more to gain more protein, although they don't show anything like the same strength of protein leverage as we do. So the net result is that you could dial up a fat healthy or a fat unhealthy mouse. You could have a lean healthy or a lean diabetic mouse. And you could do all of these things by simply playing with the protein carb fat ratio. And by dialing these things up or down, you could induce specific different phenotypes, and you could dissociate adiposity from metabolic health, for example, and demonstrating that these things aren't all of a piece. So it's nutrition and nutritional mixtures can have multiple different outcomes, and you can quantify and compare those using nutritional geometry.
Interesting. The total lifespan. So was that the same, or so the median lifespan for the high protein was less, but was the total lifespan the same or different or less?
So total as in maximum?
Yeah, in maximum. Sorry, maximum.
Yeah, the maximum lifespan and the median lifespan, both of those are measures. They're not very good measures, actually, of the actual rate, the age-specific mortality, using the proper term across the life course. And that, when we did that analysis, that required developing a new set of techniques that brought in demography, life table analyses, along with survivorship analyses. What we're able to do there is to see precisely how the risk of dying changes across the life course on different diets. And so that supersedes the median and the maximum argument. It just shows you precisely how the risk of dying changes with diet across the life course. And that showed very clearly that your risk of dying was low for much of the life course on a lower protein, higher carb diet. And then it swapped to being lowest on a higher protein, low carb diet late in life. So I guess the answer is that you see those patterns most clearly if you analyze the data in a way that looks at age-specific mortality rather than median or maximum.
Got it. So mice, grasshoppers, crickets.
Yep.
Humans, crickets. Humans. So how you've also looked at humans. Can you talk about what evidence we've seen that these same rules apply in humans? And in particular, like what kind of appetites do we have?
So let me just pick up before I'll hand to David on the human side, but the other really crucial thing, which is relevant to humans that we showed in the next set of mouse experiments, was that the quality of macronutrients matters a lot too. So we've spoken about mixtures of balanced protein, carbohydrate, and fat. What we've then done is to use nutritional geometry to unpack amino acid balance, which speaks to the question you asked earlier, Richard, but also the types of carbohydrate and the types of fat. And it turns out that a really critical interaction is between protein and the type of carbohydrate. And a low protein, high carb diet can either lead to the longest lived mice, the healthiest mice, or very short-lived unhealthy mice if the carbohydrate is extremely digestible, simple sugars. So the type of carbs matters a great deal. So this relationship between macronutrients needs to take account not only of their relative proportions one to the other, but also the composition of each of those macro dimensions. And that becomes incredibly relevant when you consider humans. And I think at that point, I'll hand to David.
Sorry, can I just have one quick question in there? So simple sugars are easily dissolved and obviously easily absorbed. And, you can have glucose and if it's something like rice, like white rice or white bread, which is simple, it's starch, but it's simple starch. And I believe we can cut it up very quickly. Would that count as a simple carb or a complex carb? White rice.
It's got a high glycemic index, so it is digested quickly and rapidly absorbed. What we did in one of our really big mouse experiments was to look at the distinction between what's called resistant starch, more readily digested wheat starch, sucrose, fructose, and glucose. So we're breaking it down from the most resistant all the way through to simple monosaccharides. And we found that by far and away the most toxic carbohydrate combination was when you had fructose and glucose in near equimolar concentrations. That was even worse than sucrose, which is the disaccharide of those two things. Now, fructose alone, glucose alone were less bad than the two combined in a 1-to-1 ratio, which is exactly what happens in the high fructose corn syrup that provides the major source of sweetener in the North American diet. So that seems to be, for whatever reason, we don't fully understand it, the worst type of carbohydrate.
Excellent. Sorry.
Which we'll get onto in relation to ultra-processed foods later and our modern food environment, I think. But in the meantime, we've discussed non-humans, we've discussed the insect studies that we did, and we've discussed in some detail the mouse studies. All of those led us to asking the question, well, we're seeing this universal thing in other species, nutrient-specific appetites and specific patterns of response when they are on imbalanced diets, experimentally confined to imbalanced diets. Well, what about our own species? So initially we did an experiment, a very simple sort of pilot experiment involving Oxford undergraduate students, where we were both based at the time. They were confined to a Swiss chalet And we manipulated their diets in much the same way as we had with the insects, and later we did with the mice. And we asked firstly, if they are given a range of foods to choose from or diets to choose from, do they also balance their intake in the way that other species do? And secondly, well, how do they respond when they are confined to an imbalanced diet with either too much or too little protein relative to fat and carbohydrates? We got a very strong signal. Firstly, they did balance their diet. They ate to a specific macronutrient ratio, like all the other species we had studied before. And when they were confined to a high protein diet or a low protein diet, they always ate the same amount of protein, but the amount of fat and carbohydrate varied. So we had answered that question in a very simple preliminary study, which the human nutrition research community didn't readily accept for understandable reasons. It was an experiment that wasn't an experiment that was done to the required standards for human nutrition experiments. There were a small number of subjects involved. There weren't extensive controls. For example, we hadn't systematically controlled for palatability in that study. So long story short, we decided to go ahead and perform a proper randomized clinical trial to the standards of human nutrition science to test these ideas. We did that first in Sydney, and that confirmed the results we had found in the Swiss study. And subsequently, we did the same research, redesigned. It was the same design of experiment, but using culturally relevant foods in a Jamaican population, and we found the same results again. Other groups have also done equivalent experiments and again found protein prioritization is what humans do. We've also done extensive studies on population data, and the patterns that we find in population studies are exactly as predicted for the protein prioritization and macronutrient selection in humans. So the answer is yes, very definitely. We have the same biology as what the other species that we have studied has, and it's not defective biology that is accounting for the obesity epidemic that we see in our own species.
And you looked at some, I guess, populations who had not got ultra-processed foods yet. They were optimizing their food intake in terms of having the right amount of protein with fat and carbs, which gets kind of dysfunctional when you have ultra-processed foods because there's not enough protein in them.
You can do that in two ways. We've done that in two ways. Firstly, you can look at a typical sort of industrialized society like our own or the United States, and you can separate out those subjects according to the proportion of ultra-processed foods that are contributing to the diet. And exactly as predicted, we see that if protein is diluted by natural whole foods, for example, lower protein plant-based foods, then you don't get the protein leverage effect nearly as strongly as what you do if protein is diluted by ultra-processed foods, which have a fundamentally different composition and are designed specifically in order to entice humans to eat large amounts of them. We can speak about what that composition is later. But we've also looked across other populations where the populations that vary in the percentage of ultra-processed foods, and you get the same thing. So protein leverage is operating. It's just that there are different consequences for protein leverage operating in a whole food environment versus an ultra-processed food environment.
And a very simple part of that is that in a whole food environment, although you may need to eat more food to get to your target level of protein, you're not necessarily eating all of the calories that would go with that in an ultra processed environment, because there's a lot of fiber and energy density is low in such foods. And we saw that in our locusts right back at the beginning. You could dilute their food with fiber and they would eat more of it. They'd get to their nutrient targets, to the same intake of nutrients, but they'd have to eat a lot more food to get there without over-consuming extra energy. So this natural foods allow that to happen, and that makes sense in that we evolved our appetite systems in natural food environments. And the other, I think, interesting aside is if you look in natural food environments in human populations that live a long time, and the famous ones include the Okinawan Japanese and some of the hunter-gatherer populations around the world that have been measured. They have a low protein, high complex carbohydrate diet, exactly as supported longest health span in our mice.
So,
and there are two reasons for that we've touched on. One is that protein, of course, is a highly satiating nutrient. So our appetites, as we've said, very closely attuned to protein, the amount that we eat, but so too is fiber. So if, as Steve said, if you dilute protein with fiber, you still get the protein leverage effect, but it's not nearly as pronounced, firstly, because fiber itself is satiating. It's the sum of protein and satiation due to fiber that determines how much you eat. Whereas if you dilute protein with simple carbohydrates, then, which aren't nearly as satiating, then we will overeat to a greater extent. And that's exactly the situation in an ultra-processed food environment. Protein is low in ultra-processed foods, partly because it's a more expensive macronutrient. So it's a commercial way of cutting the costs and increasing competition in relation to the sale of those foods, and partly because low protein macronutrient mixtures with roughly an equal balance of simple carbohydrates to fats tend to be most highly palatable in humans and also some other primate species that have been studied. And then the other reason, of course, is that what we've just said is that we are much more likely to overeat energy in ultra-processed diets. And as Steve was saying, the nature of that energy makes a big difference. And the energy that we do overeat tends to be highly purified, highly processed, simple carbohydrates with high glycemic index, exactly the kinds that we found in our mice impact negatively on metabolic health and lifespan.
Thing is that ultra-processed foods, they have lots of carbs, lots of fats, very little protein. And so we just get lots of calories.
And very little fiber, yeah.
And very little, and like no fiber, yeah.
It's very difficult to discuss human nutrition without discussing ultra-processed foods, because ultra-processed foods have become the most common dietary pattern in our species.
Yes, absolutely. Yeah. So one thing, we are also playing around with our real foods, right? So like an apple now does not look like an apple 300 years ago. Can we trust our whole foods as well? Or have they now been made so sweet that they may almost count as ultra-processed foods?
That is a question of scale rather than a question of quality, because in the wild, you get tremendous variation in the composition of fruits and other foods available to the wild primates that we've studied, for example. And despite that variation, almost because of that variation, they're able to select a very healthy, balanced diet. That is variation by degree. What's happened with ultra-processed foods is that it's categorically different type of food that is being offered, that is not available and has not in the natural world and has never been in the history, not only of our own species, but of life. And so that's a different issue altogether. So yes, overeating, for example, choosing fatty cuts of meat excessively is going to be a problem. I don't see that eating sweeter fruits will necessarily be a problem because they're always high in fiber in any case. So there's a limit on the amount that you can eat, but sweeter fruit juices might be an issue, especially ones where the fiber has been removed, because then that break has been removed in the same way as it's been removed in ultra-processed foods. So I think our appetites, to me, the evidence suggests that our appetites are well capable of dealing with increased variety in the compositions of whole foods, but ultra-processing is a different category of processing to which they aren't adapted.
And it's happened so much more rapidly. So Richard, you're exactly right in the sense that the whole of human nutritional history has been trying to increase digestibility and increase nutrient density of our foods, either by the way we process them, cook them, grow them, select them, breed them. But those things have happened at a pace that's allowed our physiology to adapt and keep up with. So we've evolved lactose tolerance or whatever it happens to be, amylase, gene duplication. But this has happened so rapidly and so extensively at a scale, as David said, that's unprecedented, that our physiology just hasn't been able to adapt.
And it really is an issue of technology outpacing physiology, because the technology has adapted very, very quickly, and there have been sort of commercial arms races in making the most palatable, the most saleable, the most profitable mixtures of ultra-processed foods. But our biology doesn't adapt over months and years like technology can. It adapts over centuries and millennia.
Yes. Yeah. We'll leave ultra processed foods for a second. So we've talked about low protein, high fat, low protein, high carb. So for humans, what does low protein mean? What is the kind of optimal percentage by calories or grams for protein?
Depends how old you are. So
Yes.
if we stick to percentages, that's an easy way to think about it. If you're a newborn, then the optimal diet is, what is it, Dave? 7% protein?
It's about 7% energy from protein, yeah, in
High
human
fat,
milk.
high carbs. Yeah. So that's indisputably the optimal diet for a human infant. That same composition would kill you in middle age, driven by protein leverage and everything else, simply because 7% protein is not optimal for later in life. So 7% protein early on. Then there are some complexities about growth and mass-specific growth rates as you go through your early life as a child and adolescent. But broadly speaking, it seems to be around 15% protein of total energy is about right, give or take. During pregnancy, it goes up a little bit. So 18 to 20% we think is about optimal. And then for much of midlife, it's probably 15, even a little lower than 15% would be what you would predict in a whole food environment would support best metabolic health during that period. And then in later life, towards your late 60s and beyond, as you become leaky with respect to protein, you start to break down protein at a higher rate and burn it unnecessarily. Then you need more. You've got a leaky bucket, as it were, with respect to protein, and you have to keep it topped up. So probably then up to around 20 or a little bit higher %. But these are not actually fully understood. Much of the recommendations around protein have been based on what's the minimum requirement and been based on measures such as nitrogen turnover rather than a more holistic ecological understanding of the evolved regulatory targets, if you like, for protein. But that's broader.
I think what that means, that trajectory that you've just described is pretty solidly established, is how protein requirements increase through the lifespan. But the level, the absolute level, is what is not that fully understood. And the other important thing is that, of course, as Steve has said, protein requirements are conditional on age, but they're also conditional on the food environment from which you source that protein. So in a whole food environment, in fact, the healthiest, the longest health span associated food environments are a relatively low percentage of protein, as we said earlier, around 10, 12, less than 15% protein. If in an Australian or an American food environment, you selected a 12% protein diet, from typical foods available, a proportion of ultra-processed foods, that will be far too low. You'd need a higher percentage to prevent from protein leverage causing excess energy intake and consequent health problems. So it's conditional on age, and it's conditional on the kind of food environment from which you source that protein.
And it's also conditional on your health status. So, for example, diabetes or insulin resistance increases protein requirements because it disinhibits the breakdown of protein. So normally, if you have insulin circulating, it's an indicator that you've got sufficient energy in the form of carbohydrate. Therefore, you want to suppress the breakdown of lean tissue because you don't need to burn the furniture to gain energy. But if that suppression of proteolysis is lifted, as it is under insulin resistance, then you'll start to burn lean tissue, subject liberated protein to gluconeogenesis, it leaks through the kidney. All sorts of things start to happen, which are essentially increasing the proportion of protein you need in your diet to not engage this protein leverage response and drive yet further overconsumption, making things worse. So there's this powerful positive feedback that can kick in as you become metabolically dysregulated.
Do you do you look at the type of protein for animal or for plant? Does that matter? Or is it the protein, or is it what comes along with it?
It's a bit of both, isn't it? So type of protein comes down to the question of the balance of amino acids, particularly. But what comes along with it is equally important. It's very hard to dissociate those two in population data. That can only really be done experimentally. And those sorts of studies in humans are very rare.
Right. So one quick question. For the energy, suppose you have high quality fat or high quality carb. Does the form of energy matter as long as you're getting your protein?
Again, do you want me to go for that one?
Yeah, you go for that, Steve.
That's a very, very good question. And it speaks to some of the controversy around what is called the carbohydrate insulin model versus the energy balance model. The answer depends on what you're looking at. If you're looking at protein leverage and appetite, all of our studies are suggesting that the ratio of fat to carbohydrate relative to protein doesn't make much difference. It's the proportion of protein relative to non-protein energy that drives how much you eat. But once it's entered your body, then that ratio matters a lot. And you can use nutritional geometry to map the consequences in terms of metabolic and other responses. And we talked about how we did that in our mouse studies a little bit earlier. So it depends what you're looking at. Protein concentration relative to all other calories is what matters when it comes to appetite control. The impact on your health and longevity is going to depend on the mixture of fat, carbohydrate, and protein that enters your body having eaten it.
But that's not well understood for humans. There's a lot of controversy around whether and how that is, which ways that is the case. And a part of the reason is it's not going to be a simple answer. It's going to be a complex answer. You need to disentangle. We've already said nutritional responses are conditional on a lot of things. One of the things they're conditional on is the food background in which the nutrients are eaten, the conditional on health status. And these are really complex things when different kinds of carbohydrates and different categories of fat interact with complex physiologies. So the simple answer of fat is bad, carbohydrates bad, fat is good, that is ultimately wrong. In terms of energy intake, as Steve said, we're pretty confident it doesn't make a difference which of those is involved, except for the fact that fat tends to be associated with foods that have a higher energy density. And energy density and protein leverage interact, because if protein leverage is a food that has high energy density, it has a greater consequence for energy intake, obviously, than if it has a lower energy density. Which is why fiber, as we said, isn't fiber-rich diets aren't a problem in relation to protein leverage in the same way that more energy-dense, fiber-poor diets, and especially fat-rich diets, can be.
Right. Interesting. Yes. And we could talk about fats for a long time. But so as we come to the end, can you talk about what is kind of the next steps in your research? What are you looking at now in terms of nutritional geometry?
Well, we're in a position now where we're able to go in two directions. We feel now that we have a really powerful integrating framework within which we've now put together enough pieces that we can see the big picture. And so where we're going at the moment is delving deeper into the mechanism. What is protein appetite? How are the amino acid signals being detected? How are their interactions with other appetite control systems and so forth? So that's an experimental program. But at the other end, and I'll hand to David now, we want to understand how we can use what we now know to contribute to rebalancing of the food system.
So it really is. We started out saying that we started our careers by inventing a more systems-focused approach to nutrition, which is nutritional geometry. Given the history we've discussed and the things we've done, we've actually got more and more into understanding systems, and the two systems that we're using nutritional geometry to probe are, as Steve said, the metabolic systems, the complexity of those, and also the other is food systems. And most importantly is because nutritional geometry sits at the interface of these things, we can understand how the one relates to the other. How does our food system impact our metabolic physiology and our health? And also, how do the mechanisms of nutrient regulation interact with that broader food system and, in fact, influence it in a way that feeds back onto the problem, either negatively or positively? And that's really important because in those two approaches, there are different ways of approaching the problem. One is through a more physiological, medicalized approach, which is very often necessary, especially given the extent to which nutrition has become a problem. But the other and the more rational approach is to deal with the cause, not the consequence. And for that, we need to look at the nature of the food system. We need to identify what is it about the food system that is driving these problems and identify the most efficacious leverage points, intervention points, where we can help to solve the problem at source rather than the consequences. And that's where we've taken nutritional geometry into the study of food systems to understand what happens in populations interacting with food environments that vary, and what are the broader ecological factors, the socio-economic and political factors that are influencing those food environments to drive the kinds of problems that we are seeing, and possibly identify solutions to those around policy, around in industry responses, and also around consumer perceptions, consumer information.
And to do all of that, we've needed a place that can engage all of the disciplines you need to do what we're talking about. And that place is the Charles Perkins Centre at the University of Sydney. So that's given us an environment where we can start to really tackle these issues at all levels in a systemic way. And I guess starting in White Woods 38 years ago and ending up here has been an extraordinary journey.
Yeah, excellent. Because it is, yeah, it's a really big challenge that you're picking, but it's so important that we move forward on that. Okay, so quickly, where can people follow your work?
I think the best is to read the book. The Eat Like the Animals is a more generally accessible account of the work that we've spoken about today and its implications, its broader implications. The other one we published together in 2012, The Nature of Nutrition, is a more academically focused, but not exclusively academically focused book. So there are different emphases. And then we have websites. A lot of our papers are available freely on the web.
Okay. Good place to start with the books.
Yeah, excellent. And we will link to those. I read the second book, the Eat Like the Animals. That was very approachable, very easy to understand.
Good.
I did have a look at the first one as well. Yes, you're more academic. Okay, so thank you so much for joining us today.
It's a pleasure, Richard. Thank you.
Thanks, Richard.
Thank you.